Hello, everybody. Uh, Welcome to the latest installment of the Torrey Society's Fall 2022 lecture series. My name is Lydia Paradiso, and I'm a graduate student at the New York Botanical Garden, as well as a member of the Torrey Council. Since the Society's founding in 1867, one of our main activities has been to host lectures on various topics of botanical research. We've hosted some great talks this fall, which you can check out on our YouTube page, which I will link in the chat. A recording of this talk will also be posted there if you have to leave early or if you want to share this talk with anyone else. The best way to stay updated on future lectures, field trips, and other activities is to become a member of the Society, which you can find out more about on our website, which I also linked. But we also post about our events on social media, specifically on Facebook and Twitter, both of which our handles are at Torrey Botanical. Tonight's talk will be presented by Dr. Andrea Fetters, who is a NSF postdoctoral fellow in the Department of Evolution, Ecology, and Organismal Biology at The Ohio State University, where she studies how below-ground mutualisms influence above-ground mutualisms. She received her PhD from the University of Pittsburgh in 2021, congratulations,
Thank you.
on ecological drivers of the pollen genome, which she will talk more of the pollen virome, which she will talk more about today in her talk, the pollen virome of wild plants and its association with variation in floral traits and land use. Before I turn over to her, quick notes about the Zoom. I did mention it's being recorded. Feel free to keep your video on or off during the talk, but just make sure you keep yourself muted so that we don't disturb the speaker. Enter comments and questions as we go. When we reach the Q&A portion, I will read out those questions, or if you want to ask your questions out loud, you can use the raise hand feature or make a note in the chat and we'll make sure that you have your chance. All right, and now I will hand the mic over to Andrea.
All right, thank you very much, Lydia. I'm going to attempt to share the screen this time.
Hope you'll remember the choice.
Yeah. Okay, are you seeing the slideshow? And okay, perfect. All right. So thank you, Lydia, for the introduction. And as she mentioned, this talk will be focused on my dissertation research at the University of Pittsburgh that I did in Teal and Ashman's lab there. But first, before I get started, I want to thank everybody for listening. And I also want to thank all of the co-authors that you see listed on the first slide, because this work was truly a collaboration and would not have been possible without the entire team, for sure. Hold on. Slight technical difficulties. Okay. You finally got it to work. Okay, so in this talk, first I'm going to walk through a broad introduction of the pollen virome and possible ecological drivers of it. And what I mean by ecological drivers are things that might influence the size of a plant's pollen virome. So that might be something like floral traits or the land use and the environment surrounding plants. Then I'm going to give a brief overview of the methods that we used to answer the main questions of our project. And then I'm going to answer the main questions of the project, which were, number one, what viruses are associated with the pollen of wild plant species? And very closely tied to that is, what is the host range of those viruses? Number two, do plant traits influence the pollen virome? And number three, do landscape traits influence the pollen virome? Then at the end, I'll tie it all together and summarize our main results and tell everyone how our work has filled in some of the knowledge gaps that currently exist in the world of pollen-associated viruses. So first, the introduction. What is the pollen virome and possible ecological drivers of it? So first of all, viruses are ubiquitous. They infect nearly all life forms across the animal kingdom, the plant kingdom, and even the microbial kingdom. But what is the virome? Before we get too much into the pollen virome, and how much do we really know about it? So the virome in general can be be defined as all viruses associated with an organism. It's hypothesized that there are about 1 × 10⁻31st power viruses on Earth, and most of those are undescribed or maybe not even related to the ones that we've discovered so far. Many of the ones that we do know about, though, are important to our livelihoods because they cause diseases like influenza, COVID-19, or actually AIDS, and then COVID-19, which can all make people very sick. And if we zoom in from the entire virome and think about only the plant virome, these are all viruses associated with the plant. And there are about 1,500 described plant viruses, and most of these are vectored by herbivorous insects like beetles, aphids, things like that. And similar to those that cause human disease, most plant viruses have also been studied in agricultural or economic contexts because they severely damage crop plants and limit yield, which then can impact humans quite a bit. We zoom in even farther and think about the pollen virome. This would be all viruses then associated with pollen grains. So this would mean like all viruses either on the outside or the inside of pollen grains too. And these are vectored by pollinators like bees as they forage for floral resources like nectar and pollen. There are just over 100 described pollen-associated viruses and viroids so far. And again, these are mostly studied in agricultural contexts because a lot of these viruses can have pretty drastic impacts on fruit production in plants. So because of that, past research has focused a lot on transmission between individuals and how certain viruses infect susceptible individuals. So as I mentioned, we know that vectors or that pollinators are the main vectors here. And these can include bees, but many other things like flies, butterflies, anything that transmits pollen from one individual to another. And plants can't walk around and infect each other like we can, so they need this type of vector to spread the virus. These pollinators, as they forage for floral resources, they transfer pollen from anther to stigma. And if the pollen is infected, then they're also transferring viruses from anther to stigma. And the stigma is a highly receptive, extremely vulnerable surface of a plant. So it's like ripe for infection. Not only could pollinators transfer these viruses as they are pollinating a plant, but they could also bring infected pollen in contact with a wound in plant tissue, or also make wounds themselves as they are foraging for floral resources, and perhaps also transfer infected pollen to a plant that way. So past research not only focused on transmission, but also, like I said, actual infection of individuals. So one common infection route is vertical infection. And so here, the virus would move from an infected parent, and in this case, the father, to the offspring housed by a healthy mother. So as if that infected pollen lands on a stigma, and if those pollen grains germinate and a pollen tube grows down, the infected sperm within that pollen grain can then infect the healthy egg within the ovule if that fertilization is successful. Doesn't always work though. The other common infection route is horizontal infection. And here that a plant can become infected if that infected pollen lands on a stigma and that pollen begins to germinate and pollen tubes begin to grow, because then the stigma or even the style can become infection sites as that infected pollen is growing. And in this case, it infects the mother plant or the other like individual plant, not the offspring. Another horizontal infection route is if that infected pollen does fertilize a healthy ovule, which then becomes infected. But then in a couple of cases, some viruses have been shown to go from like that tissue that's connecting like the developing embryo still with the mother plant, and the virus can travel back and forth through that avenue. And then the last way that this could possibly happen is if infected pollen does contact a fresh wound in the tissue, either made by the pollinator while it's foraging or just on another wound there. And this might be especially true if the virus is on the outside of the pollen grain instead of the inside. And I think pollen-associated viruses are really cool because as you can see through all of these infection routes, like some pollen-associated viruses can infect more than one generation in a single like point of contact, whether so infect both susceptible offspring or the mother plant as well. So okay, so there then we know quite a bit about how infection by pollen-associated viruses occur and how they even get to a plant. That information is really important. We don't know a lot about the ecological drivers of the pollen virome. And these could include things like virus traits, plant traits, pollinator traits, and even traits of the landscape in which these plants are growing. So that you can imagine that there are probably endless questions one could ask about the system. For this project, we focus specifically on plant and landscape traits. So for some of the possible ecological drivers, first think about some plant traits that could influence the pollen virome. Sorry. More specifically, these include flower traits that mediate interactions with those pollinator vectors. For example, maybe having multiple flowers allows a plant to interact more with pollinator vectors and also have increased numbers of plant pollinator interactions, and maybe therefore more pollen-associated viruses. On the maybe flip side of that, if you have maybe fewer flowers, but those flowers have a more closed flower morphology, maybe those flowers have more intimate directed interactions with those pollinator vectors. Maybe those flowers receive more pollen, and maybe it's more likely that a pollen-associated virus would come in contact with that plant host because of the tight interaction with the pollinator vector. Lastly, pollen traits are really important for pollen grain collectability and transfer by pollinators. So it's also possible that having something like a spiky outer layer or a spiky pollen grain exine might lead pollen grains to be more easily transferred between plants and cling more easily to pollinator vectors, therefore increasing a plant's chances to have pollen-associated viruses. Another of the many possible ecological drivers of the pollen virome are landscape traits. Some landscapes are heavily fragmented by human land use, while others are more isolated and have very little to no human land use, while many other areas lie somewhere along the middle of that continuum. One example of human land use is agriculture. Viruses often emerge and thrive in agricultural environments because potential plant hosts are packed so closely together. And crop plant species might also share pollinators with wild plant species, increasing more potential transfer between groups of plants. So to recap all of that information that we just went through kind of quickly, past research has focused on pollinator or pollen-associated virus transmission and vertical and horizontal infection routes with like 1 or 2 viruses per study, focused on like 1 or 2 agricultural plant species per study. So even though we have a lot of mechanistic knowledge, we're missing kind of the bigger picture of the pollen virome, I think. And which is why we wanted to ask, number one, what are viruses associated with the pollen of wild plant species and what is their host range? Because as I mentioned, a lot of those studies have been done in plants of agricultural or economic species important, almost nothing about wild plants. Number 2, do plant traits influence the pollen virome? And number 3, do landscape traits influence the pollen virome? So the methods that we use to answer these main questions are a variety of field, lab, and computational methods. First, I'll tell you a little bit about the plant species themselves. So this project was a survey of 24 plant species spread across 4 different landscapes in different regions of the United States. These plant species are all herbaceous, animal pollinated, and most were perennials. The landscape trait that we focused on for this project was human land use, most broadly, and more specifically, we focused on the amount of agriculture and impervious surface in an area. So those 4 landscapes ranged quite a bit in the amount of human land use that they had. This first, we called the Eastern Deciduous Agri-Forest Interface, which is heavily fragmented by agriculture, and is characterized by 56% of human land use. So this GIS picture here shows, and other that I will show like it, show natural vegetation in green, impervious surface in red, and then yellow is agriculture. And then another region that we surveyed was the California coastal region. This is characterized by like dunes, scrubby vegetation, some urbanization, but characterized by only about 12% human land use. The Central Appalachian region that we surveyed is characterized by mostly preserved habitat, and we stayed mostly in the Blue Ridge Mountains for this, but it still had around 11% human land use. And then lastly, the California grasslands region was the 4th region that we surveyed, and it is dominated by very remote areas of chaparral vegetation and serpentine seeps that are really important to the communities out there, and it has only 2% human land use. So 2 sites more on the extreme end and 2 that were a little more intermediate in terms of human land use. So the plant species that we surveyed in this project differed in a lot of pollen, grain, and floral characteristics that we chose to zero in on for this project because they're important to those plant species interactions with pollinators. And it also influence pollen grain collectability. So the flower traits include flower size, inflorescence type, so whether it's a single or a multiple flowered plant, the flower longevity or how long these flowers live, along with the rewards that they present to pollinators. So whether it's either nectar or pollen, or it has both nectar and pollen. The flower symmetry, and also very closely related to that is the reward accessibility of those, of either the nectar or the pollen or both. And then the pollen grain traits that we thought about were the pollen grain texture. So does it have a more smooth outer layer, or is it more spiky? And then how big are those pollen grains? And all of our hypotheses related to these plants basically were that plants that have traits that increase plant-pollinator interactions and pollen grain collectability would lead to a plant species having more pollen-associated viruses. So for example, bigger flowers, a plant with many flowers might have more pollen-associated viruses than a plant that has only one flower that might be smaller. So for this work in the field, we collected a lot of flowers. And then we sonicated the anthers of those flowers so that we collected only the pollen. And I want to stress that we did this in as sterile a manner as possible in the field, which is hard. And we also removed any plant tissue samples or other debris that got into our pollen samples so that we were getting only pollen in those samples. Then once we brought it back to the lab, we freeze dried the pollen samples for at least 12 hours because pollen is a really resilient, hard to break product. And well, we wanted to break the pollen grains so that we could get an idea of the viruses, not only on the outside of the pollen grains, but the inside of the pollen grains. So after we freeze dried the pollen, we then broke the pollen apart and then checked under the microscope to make sure that, in fact, the pollen grains did break. Then we extracted the total RNA from those samples because most plant viruses have RNA instead of DNA genomes. Then we sequenced that RNA, and we collaborated with a virology lab at the University of Pittsburgh who had developed their own viral detection pipeline called Pickaxe. And from there, we could figure out what known and novel viruses were in each of our pollen samples. And in the next couple of slides, I'll talk a little bit more about what I mean by known and novel viruses. So to identify the known and novel viruses, we use this pipeline called Pickaxe. And after some initial quality control and like host genome subtraction steps, we are left with these pollen-associated virus or non-plant reads. We were then able to extend or assemble those reads into contigs in a lot of cases. And then we aligned both the reads and the contigs to various databases in NCBI, where a lot of reference genomes are kept so that we could figure out like what was actually in our pollen samples. So we wanted to be, more confident about what we were identifying and making sure we weren't just picking up things by random chance or tiny fragments of things that maybe weren't really there. We did impose some selection criteria for each of these, for the known virus and the novel virus identification. So for a known virus to be like truly present in our sample, we wanted to make sure that those reads aligned at least 10 times to a reference genome and covered at least 20% of that reference genome. And then for novel viruses, this is where we use the contigs. And overall, we were trying to see if they were less similar to a reference genome than species demarcation criteria defined by the International Committee on the Taxonomy of Viruses. These rules are different for every virus family. So we had to go in and see like where was that percent identity threshold basically, and were our contigs more or less similar to a reference genome than those cutoffs. But not only did we have those cutoffs, but we wanted to see like, okay, so if we have a large contig, does it encompass all of the viral proteins that might be encoded by a putative virus family? So we have 4 flavors of novel viruses in this project. The first we called the novel coding complete viral genomes. And this is where like, was the contig long enough to correspond to a putative viral family? Were the viral conserved domains or proteins, were those all present or mostly all present in the contig? And was that percent identity less than that of the rule set out by the ICTV for a given family. And then like very similar to that, but a little bit different, were the novel coding complete strains of known viruses. Same criteria as above, but the species, but the percent identity between our contigs and the reference genome was higher than that species demarcation cutoff level set forth by the ICTV. So these were too closely related to a known virus to call it completely novel. And then we also had these novel partial viral genomes and novel partial strains of known viruses. So same exact criteria as before. However, these contig lengths were much too short for us to really confidently say, you know, oh, these belong to a certain virus family or not. However, we did take notice of these RNA-dependent RNA polymerase proteins whenever we did see them, because these segments are used for virus identification in many studies. They're species specific. So we are pretty confident that if we saw an RDRP, it was indication of another virus truly being there. And then we put all that information together to really quantify the pollen virome. So we have two estimates of pollen-associated virus richness here, and one of them is more conservative. The first one was just that total number of known viruses plus the number of novel coding complete viral genomes and the novel coding complete strains of known viruses. The relaxed estimate included everything in the conservative estimate, but also those RDRPs, those like partial RDRPs that we found. Okay, so now that I told you about how we do this, finally going to answer some of those questions. So, or not some of those questions, all of them, I apologize. But anyway, number one was, what viruses are associated with the pollen of wild plant species? And very closely related to that is, what might their host range be like? So I'm using this presence absence grid to illustrate the answer to this question. And the plant species from the survey are along the top. The viruses, the known viruses and the coding complete novel genomes, both fully new and strains, are along the left, and then the blue squares correspond to the region in which the plants and viruses were found. So we found 22 known viruses. Those are underlined in blue, 10 of which were found in more than one plant or landscape. And those are denoted by the purple stars there, which look a little bit out of line. So I apologize with that. Actually, way out of line. Sorry. And then we also found 6 novel coding complete viral genomes and 3 novel coding complete strains of known viruses. And those are underlined in orange there. So then our second question was, do plant traits influence the pollen virome? So I mentioned before that the 24 plant species that we surveyed for this project exhibited a lot of diversity across flower and pollen grain traits in this project. And that's simply what this PCA is showing here. And the dots represent each plant species. And the most important traits for PC1 were the number of flowers in an inflorescence and flower symmetry and reward accessibility. The most important traits on PC2 were pollen grain size and then pollen grain texture. We then used those two PCs along with PC3 together, which explained about 65% of the variation in the floral and pollen grain traits as predictors in linear models to explore whether these traits really do influence the pollen virome of the 24 plant species surveyed in this project. So in this graph, you're seeing PC1 across the x-axis, which larger values reflect multiple flowers, bilateral symmetry, and restricted access to floral rewards, and smaller values reflect single flowers, radial symmetry, and unrestricted access to floral rewards. And then on the y-axis, you're seeing the the log transformed virus search, conservative estimate of virus richness. And here you can see that multiple flowers, or having multiple flowers and bilateral floral symmetry and restricted access to floral rewards led to a plant having richer pollen viromes or more pollen-associated viruses. Here you're seeing a very similar graph, except for you're seeing PC2 across the x-axis, and here larger values mean smoother, bigger pollen grains, and smaller values mean spiky, smaller pollen grains. So as you can see that the smaller plants with spiky, smaller pollen grains had more pollen-associated viruses relative to plant species with smoother, bigger pollen grains. Then our last question was, do landscape traits influence the pollen virome? And just as a reminder, in this project, these landscapes varied a lot in the amount of human land use, specifically that impervious surface in agriculture. So here in the stacked bars next to each landscape square, you're just seeing, again, the breakdown of the natural vegetation in green, the agriculture in yellow, and then the impervious surface in like the orangish color. And the California grasslands had the least amount of human land use with 2%, while the eastern deciduous agroforest interface had the most with 56%. So here, kind of a similar graph to what you've seen before, with the conservative estimate of virus richness along the y-axis, but here on the x-axis, you're seeing each of those regions with the amount of human land use at the bottom. And you can see that plant species in landscapes with more human land use had richer pollen viromes, with plant species growing in that eastern deciduous region having the most, and plant species growing in that natural vegetation dominated landscape in the California grasslands having the least amount of pollen-associated viruses. So in summary, we identified a lot of pollen-associated viruses. I know like 9 or so doesn't seem like a lot for the novel viruses, but it was really cool to do this in pollen because, especially in wild plant species, because no one has done that before, and to identify many known viruses. And then if you think about all the little RDRP regions that we included in the relaxed estimate, there were a lot of viruses identified, and we did see that several of them, like over 10, were in more than one plant species or more than one landscape, which speaks to the fact that maybe the host range of these viruses can be broad. I know this is one study, one snapshot in time, but if the host ranges of these pollen-associated viruses can be broad, then that could have some implications for conserving wild plant biodiversity and also food security as well. And then we also showed for the first time that plant traits influence the pollen virome. So plant species with flowers mediating more, but also more like intimate directed interactions with pollinators and pollen grain traits that are important for pollen grain collectability have richer pollen viromes. Smaller pollen grains, spikier pollen grains might be more easily collected by pollinators, and spikes might allow them to, cling to a pollinator more, like on hairy bees and also on plants themselves, like that stigma surface. And then lastly, yes, landscape traits definitely influence the pollen virome, at least plant species growing in landscapes with higher amounts of agriculture and impervious surface have richer pollen viromes. And I just want to again say that there are many unexplored ecological drivers of the pollen virome. I'm sure there are many other plant traits and landscape traits that could be thought about, and as well as many virus and pollinator traits, and also the interplay between all of them could definitely be studied to really explore the pollen virome and what that might mean for host plant species and pollinator vectors, and something that I hope to go back to someday. And with that, there were many people to thank for the success of this project. Again, my co-authors, and then my dissertation committee at the University of Pittsburgh, the past and previous members of the Ashman Lab that I was fortunate enough to interact with while I was at Pitt, and then the many field sites and facilities that we traveled to collect this pollen. And I would like to thank you all again for listening, and I'm happy to take any questions you might have.
Awesome. Thank you so much. I know I was really into, when I first kind of read the title of the paper, the first time I saw it, I was like, this is a really cool topic that I know I never really thought about. And especially, that viruses, in recent times have become, are in people's minds. A lot of people are learning more about like how viruses reproduce and all this kind of stuff. I think it's really cool. So yeah, if anyone has any questions, you can put them in the chat. You can raise your hand. I see we got Amy is ready. You can go ahead and ask your question first.
Okay, I have a lot of questions, but I'll just start with one so other people have a chance. And this is at your landscape level. Did you find that when you were in the area with more, the Eastern deciduous agricultural interface with more human interface, were you getting your better known viruses there? And were you getting more poorly known viruses in your location with less human interface?
Yes, that is exactly what we found. And we all think that it could be the fact that, I haven't really done this, but other people who have studied pollen-associated viruses have, looked at species of agricultural importance, or maybe even plant species, that are constantly around humans too, that are easier to get to, could have been sampled more. So yeah, for sure. That's what we found.
Yeah, thank you. Very interesting talk.
Thank you.
All right, Brenda, you can go up next.
Awesome talk.
Thank you.
It's a new concept. I learned a lot. I have a quick question about, can you trace the origin of the viruses?
I don't know. I'm not sure. Like the way we did it, probably not. And even if you're just going out and collecting pollen samples, I'm not sure if you could really figure out where they're coming from, or like what plant they started in. Like, for example, if the viruses that we found, like if they were from an agricultural plant species and now they're in the wild plant species, or if they went, vice versa. It's a very interesting concept, and definitely, raises a lot of question of like virus spill over and spill back, like which way are these moving, and what could the impacts be for both of those groups of plants, depending on the direction of those arrows. One limitation of this study is definitely that we also didn't know, or couldn't even know, like what these viruses were doing to the plant host, if anything. We don't know if they were actually actively infecting them and causing problems, or if they were just simply there.
That was going to be my next question.
Yeah. Yeah,
Thank
we
you.
don't know. Unfortunately. Because they, by eye, none of these plants looked particularly sick beyond, just the normal wear and tear that you see in the environment anyway. We didn't ever see any, hallmarks of like viral disease, which are like mottled leaves or
Yeah, I was going to ask you, yeah, like
spots.
mosaic patterns or that kind of stuff.
No. So they were all, to our knowledge, asymptomatic, but that again is limited by what we could see just when we were out there collecting pollen.
It will be interesting to see if they can actually affect pollen tooth
Yeah.
growth and competition among the
Yeah.
different pollinators. Anyway, thank you. It
Yeah,
was awesome.
you're welcome. But I think they can though. I think there have been some studies that have shown pollen sometimes doesn't grow if it's infected. Sometimes it does. That's why fertilization by infected pollen is not perfect, because
Okay.
it impact different viruses in different plant species have different impacts.
Thank you.
You're welcome.
We had another question just like that in the chat about whether these were they problems for, I guess, the plants or the pollinators. But it's also interesting because I was thinking because this has been only studied in certain contexts, there could be these asymptomatic infections, or they're, dormant, kind of, or latent infections, whatever, that you don't have those classical signs. And that's.
Yeah. And to answer the question about were they a problem for pollinators? Well, we found deformed wing virus in quite a few of our pollen samples, especially from the eastern deciduous agroforest interface area. And that is very detrimental to bees. So, but it's commonly transmitted in pollen too. So, maybe that was, yeah, that was there. That was maybe to be expected. There was an interesting study published several years ago now. I forget what year it came out, but they showed that a plant virus was actively replicating in honeybee tissues, which so maybe leads, or begs the question, can these viruses eventually jump from, one kingdom to another? And maybe they can, but we don't know. The authors of that study, they don't really know what it was doing to the bee, if anything. They just found it there replicating, but it's interesting.
Definitely. We have two related questions in the chat from Marielle and Amy about the choice of the plants that you were using. You could talk.
Yes. So we chose plant species in each region based on a couple of different things. One was how many individuals were there, because we collected, we aimed to collect 30 to 50 milligrams of pollen per plant species, and that's a lot of pollen. And so we needed numbers, and we also did try to, once we got a plant species of one family, we tried to stay away from that plant family as much as possible and try to get a diverse collection as possible. So I know in the eastern region, we did have a couple asters in the same, in that sampling block, because that was also what was available when I collected pollen there in August. But we tried to do it that way, but also, thinking about okay, what plants here are providing resources to pollinators too, which is why we stuck with these herbaceous flowering plants and didn't use things like woody trees or anything, for example, although those do provide resources to pollinators.
Yeah, sometimes it's that the necessity part of things. It's that's what there was, and
yeah,
that, I mean,
exactly.
I don't know how many. I guess it also depends on how much pollen they're producing, but that sounds like, it doesn't sound like a lot until you realize how light and small pollen is.
Yeah, yeah. Some of the flowers, they were not hard to collect pollen from, and it went quick. The sampling went quickly. Others, not so much. And we were collecting pollen from a single plant species for like an entire day to get the amount that we were going for.
And that machine that you were using, how big and heavy was that to have to carry around?
It actually wasn't that bad. It was a like portable sonicator, and it really was only, I think, like this long, roughly this long, and it came with its, a little like, shouldn't say docking station, but kind of like, to hook the probe up to like how fast we wanted to make the sonicator work, and then carried around a portable like battery generator to plug that into in the field. And I mean, it worked out. It wasn't that bad. We did have a lot of things to carry, but in most places it wasn't a huge problem.
So you didn't have any rainforest samples, so you didn't have to
Right.
through the.
Mm-hmm.
All right, Amy, you have another question?
Yeah, I've always got lots of questions. So I may have missed this, and I suspect I know the answer based on just the amount of pollen you had to collect. But did you do any unsonicated pollen to distinguish what was on the exterior of the pollen grains from what might be inside the pollen grains?
Well, we did not do this unsonicated versus sonicated, but and to my knowledge, I don't think the sonication itself broke the pollen grains all that much. I mean, I could be totally wrong about that, but we then, after we brought the pollen back into the lab and after we freeze dried it for a long time, we did break the pollen sample because we were attempting to see the pollen virome, not just on the outside of the pollen grains, but the inside of the pollen grains. However, it would be really cool someday to do a project where you do where you look just on the outside versus inside, or what's on both.
Or take your pollen sample and divide it into two treatments, and then break one and don't break the other, and that might give you a way to get at that a little bit more.
Yeah, that would be really cool. And the amount of pollen that we used, like we did a lot of troubleshooting with things that were just growing in the pit greenhouse actually before we went out into the field, and we got the most reliable RNA extraction results from 30 to 50 milligrams of pollen, but we did get some results sometimes with as little as 10 milligrams of pollen. So it depends, and I think it's definitely doable to do answer a question like that.
Yeah, very cool.
Thanks.
Brenda, go ahead.
It's awesome. Do you see like a relationship between seasonality and the diversity of the viruses? I'm thinking like, winter, flu season, like that?
That would be a really interesting question, but I have no idea right
Okay.
now.
Well, because I wonder if that's not so much about the viruses being more around, it's that we're more immunocompromised in the winter. Maybe that's why, but
Yeah,
plants too would theoretically also be more vulnerable in the winter.
yeah.
So,
So yeah,
Or
that
during stress, like drought situations. Okay.
Thank
you.
Thanks.
I have also one more thing about the viral taxonomy.
Yeah.
It's very interesting because I guess especially when you have these viruses, there's not a lot of morphological characters to go on or that they can readily observe. So it's interesting that they have this like gene percentage cutoff, because that's such a big question in like, I guess all kinds of genetics of like, can you quantify what's a different species versus what's a variety or a strain? So
yeah, I think among viral taxonomists, that is a very hot button issue about what is actually different versus what is not. And to maybe get around some of that, the ICTV also has, like, in addition to these species demarcation criteria, like this percent identity criteria, they also have other things that they look at, like what does it do to the plant host, or like a host, like what does a virus do, oh man, I should know, but there's like a lot of things, like what I'm sorry, I'm forgetting them all right now, but there are many other things that they look at beyond just percent identity of these conserved domains or these proteins. Because of how we were doing our project, the species, the percent identity between the genes were about the best we could do. That's not to say there's not other things, because there is.
Very cool. Yeah, it's interesting the similarities and differences of the different like kingdoms.
Yeah.
And when it comes to that kind of stuff.
Yeah. I think it's in flux a lot too, because between like when we, the first time we submitted this paper to the time it got accepted, like I think, families were different, other things were shuffled around, like, a new family was described for a virus that in our list of viruses like previously didn't have a family name assigned to it. So I think it changes often too.
Yeah, exciting field
Yeah.
of virology. It's
Mhm.
especially if there's this like enormous crazy diversity that exists out there.
Mhm. Yeah, there is for sure.
And their generation time is much faster than ours, so it's going at a
Oh yeah,
accelerated rate.
extremely fast. Yeah.
Well, if anyone has any last questions that they want to ask, we can do them now.
Mhm.
Also, actually, Andrew, if you want to throw your email into the chat,
I can happy to do that.
if anyone comes up with anything else that they want to ask, you can reach out to her there.
Yep. Just there's my Ohio State email, and I look at it all the time.
I have a quick question.
Mhm.
I'm not sure if I looked at the chart right, but did all of your plant samples have viruses? Did you find viruses in all of them, or did some of them not have?
Some of them did not have any viruses, and most, if not all of that, was from the
lily.
California. Yeah, was in the California grassland region, which had the lilies, had an iris. I'm gonna look back at that really fast just to make sure.
Yeah, I wasn't sure if I was looking at the chart wrong, but it was like, well, that looks like that whole group doesn't
Oh
have.
yeah, that whole group didn't have anything, and all of those plants, well, not all of them, several of them were from either of the California regions. And then one of them was from like the Central Appalachian region, that Trillium. And then the other one that didn't have anything in it, the Thermopsis, also from one of the California regions. So mostly if we didn't find a virus in a plant, it was from one of those California regions.
And were those species that you were able to get large amounts of pollen from, or smaller amounts?
Definitely large amounts, or we got to the desired amount
What you
of
wanted,
pollen
yeah.
very fast compared to some of the others.
Oh, cool.
Yeah, which was always a breath of fresh air when you didn't have to spend hours collecting from one plant species.
Also, how big are the viral genomes about?
Only probably on average like a couple thousand base pairs or less. Really not very big.
Yeah.
Which is why it was really exciting when we could assemble those reads into contigs that had the required number of base pairs for a putative virus family, and then after doing basically just copying and pasting those sequences into ORF finder, which tells you where are the proteins in here, or where might there be proteins in here, and then taking those ORFs and those sequences of amino acids and putting them into a conserved domain database search. So seeing okay, if we take this protein sequence, does it actually hit anything meaningful? And when it did, and when it was the correct proteins in the correct order for a given viral family, that was super exciting to us.
That's really cool.
Thanks.
Thank you.
I'm a little jealous as studying something that has a 12 gigabase genome.
It's huge.
That sounds great.
Yeah, viruses are nice sometimes.
At my school, we study bacteriophages, with our students, and those are even smaller than yours.
Yeah. Yeah, they're tiny.
Very cool. Thank you.
Yeah, thank you.
All right. Well, yeah. Thank you, Andrea, so much for this talk, and thanks everybody
course.
for coming and for your questions and your interest.
Mhm.
We're always happy to host these and give people an opportunity to learn something, give their talk, and everything like that.
Yeah, this was a lot of fun. Thank you for the invitation.
Yeah, totally. All right. Well, everybody, have a great night. Stay well and have a happy Thanksgiving. And hopefully we'll see you again soon.
All right. Thank you. You too.